everyone. Thank you so much for joining us and appreciate your patience as we're getting started here. For those who don't know me, my name is Chris Lustrino, founder and CEO of Kingscrowd. This is a series that we run for our members here at Kingscrowd, as well as those folks who are interested in Siren or are already investors in the business. And essentially, this is an opportunity to get a deeper dive look at why Kingscrowd Capital invested in this business, the fundamentals that we were excited about in looking at this organization, and then also be able to get a deep dive with the founder themselves and really get to know Nicole better and understand more about her background, what they're working on, and get your investor questions answered live in a more intimate setting. So everything we do, right, is trying to educate and empower individual investors to be able to make smart, informed investment decisions. And hopefully this is just another piece of content and access to the founders that helps you make that decision. So with that in mind, I'm going to invite Nicole to come on up here in a second. So feel free to come on up, Nicole. But in the meantime, just wanted to hit a little bit on, some of the things that we love about Siren. We have looked at hundreds and hundreds of biotech deals over the years. And Siren, for me, as only my opinion, one of the most exciting, interesting biotechs that we have looked at, just from the profile of the technology that they're creating, what they're trying to solve for in brain cancer, an acutely challenging area of cancer that has certainly not been solved at all, and is creating something that I think is, in the best in class category. There's no guarantees in any of this, especially with biotech, early stage biotech, but they're doing a lot of the right things and have brought on an incredible team and investor network. So this is a really, really interesting deal, and I'm excited to get into it here. So Nicole, thank you so much for joining us today.
Happy to be
here. So for those who, don't know as much about Siren, dive in, tell us a little bit about your background, how it came to be, and what you're all about.
So this is actually work that spun out of my lab. So for folks who kind of don't know me or didn't know me before, so I'm Nicole Paulk. I'm the CEO and founder of Siren. And prior to this, I was actually a professor of virology of AAV gene therapy at UCSF here in San Francisco. And this was work that kind of got inspired by work that was happening in my lab, and we decided to spin it out at the peak of COVID. And when I say the peak of COVID, I mean like the peak. Like, remember when you were still like Lysoling off your food that you had got Instacarted to your door, and then you waited 10 minutes to open the door before you went out and got it? Like the peak of COVID is when we spun out. Which was an interesting time to be a virology professor too, by the way, but a separate story for a separate day. And this was all motivated based off work in my lab, where I was a fairly unique professor in that I was a gene therapy professor that didn't work on gene therapy. What the heck does that mean? It means we were working kind of like at 40,000 feet in the clouds on like the concept of gene therapy itself, like using viruses to treat patients and deliver genetic medicines, but we weren't working on any particular disease. So we were just working on enabling technologies to make this class of medicines cheaper, easier, and faster, and like easier for patients to access these types of things. And like, you name a company in the gene therapy space, I'm probably advising them in some capacity, either on their scientific advisory board or as like an external KOL that gets brought in when they go on CMC hold or clinical hold, or an hourly, advisor that's helping them through some challenging moment, these types of things. And I kept getting asked to do things where, man, it was just like we kept running into the same problems. And I thought my lab could perhaps address some of those. And one of those problems that we kept coming up to was the fact that the particular virus that we use is called AAV, but the nuance doesn't matter. This particular class of viruses, AAVs, man, everyone kept making a brand new one from scratch for every single disease they were going after. And not only were they making like the virus from scratch, but all of the component parts that go into making a virus, they were making from scratch as well. So like, everyone has seen, even if you're not a virologist, you don't know anything about viruses, right? You've all seen a picture of like a COVID virus on like a news story or something, right? It's this like icosahedral protein, right? And, it might have little spikes on it and stuff like this. And it's carrying something inside of it, or it's carrying a genome, it's carrying a payload, some cargo. And when you make one of these viruses on purpose, right, in order to like treat a genetic disorder, when you're going to use it as a medicine, you've got to make that outer protein shell. You've got to make all the component parts that get packaged inside of the virus. You've got to make like the solution you're going to dilute it in. You've got to like come up with like the delivery device and the route of administration, like all of these things you have to innovate. There's like dozens and dozens of like components that go into making a virus. And every one of these companies that I was advising wanted to own, of course, right? We're all obsessed with ownership. We want this IP moat. Everyone wanted to make and own every single one of those component parts that goes into making a virus. It's like saying, I want to own this car company, and we want to own not only like our brand, our logo, but we're going to make from scratch the engine. And like we're also gonna make the rubber for the tires. We're also gonna make the glass for the windshield. We're also gonna make the leather for the seats. We're also gonna make like the catalytic converter. And we're also it's just like and it's just like it's really hard to be an expert in all these component parts. And so what ended up happening is that you would get these companies that wanted to produce every single component part of these viruses from scratch, own it all. And lo and behold, fast forward, what ends up happening, right? You get some very expensive drugs at the other end of that. The 5 most expensive drugs on Earth are all viral gene therapies. They are $1, $2, $3, $4, and $5 million per dose. They are the 5 most expensive drugs on the planet. Fun fact, most people probably don't know that there are 5 FDA-approved viruses you could go get right now. There's actually 7. There's 7 FDA-approved viruses right now, where like you could get sick, you could get a prescription for a virus, they can treat you with that virus and then treat you and in some cases cure you of that disorder. So like let's start from there, right? That is the concept. And so what Siren wanted to do, which we started in the lab before Siren even existed, was to address this head on. Like, could you make, rather than having to innovate every one of the component parts that goes into making a virus, including the actual medicine, like the payload that you're delivering with the virus, what if you could make a universal gene therapy that could be used to treat more than one thing and where you did not need to change out all of the component parts when you go after a new disease, right? If you're Porsche and you are making like your second car, it'd be really nice to be able to reuse like the engine from one car into another car, and all you're innovating is maybe like the body style. But it'd be really nice to be able to like keep the chassis, keep the axle, keep the engine, keep the wheels, and you're just engineering like a new body style that has like a different look, right? But what if everything could be reused, including the medicine itself, the payload, the cargo that goes inside the virus? And so we wanted to see like, just like conceptually, could you make a universal AAV gene therapy that could be used to treat more than one thing? And if so, how would you do that? And 2, could we start pointing the ship at larger indications with many more patients? Historically, we've always used these viruses to go after very, 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 very rare, what we would call ultra orphan indications in children. And we were like, well, what about cancers, right? Cancers are like this area with huge unmet needs, which are still rare, by the way. They're still rare diseases. They're just rare in a different numerical category. And so we wanted to kind of both be the first AAV gene therapy to really come and bring this modality into the cancer space for the first time, and also to make the world's first universal gene therapy where you could use it to treat more than one thing. And if we could do that, then like now you could bring the cost of production, the cost of innovation, the cost of implementation, the cost of delivery, the cost of all of these things down dramatically, so that you don't need to have a drug that is $5 million per dose. You could have something that is much more in line with kind of normal drug prices, normal drug accessibility, these types of things. And so we began that project, and then fast forward ended up spinning it out into Siren, and I loved it so much, decided to resign and actually go and be the full-time CEO, and have been doing that ever since. So that was back in 2021, and now we're off to the races, and we're going to be in the clinic here hopefully in early 2026. So it's about to get real.
Yeah, well, let's talk about, well, I'll take one step back. In terms of what your viral gene therapy can do, or it's not a gene therapy, right? It's just a virus. It is gene therapy. Okay.
It is still a gene.
Basically, can you explain what it does when it goes into your body? How does it go to the brain, and then tackle those brain tumors?
Exactly. So, fun fact, depending on your perspective, every one of us has a tumor right now. You've actually had a tumor every single day of your life. But don't freak out. You don't have cancer. You have a tumor, but you don't have cancer, right? You have tumors all over. Like I have a tumor here and here, and here, but they're individual cells, right? You've had a tumor you were born with a tumor, right? You've had individual tumor cells throughout your body your whole life. The reason you don't have cancer right now is that your immune system is actually phenomenally good at sniffing out and finding these cancer cells and chewing them up and destroying them while they're still individual tumor cells. It's this process that like nerdy scientists like to call basal background cancer immunosurveillance, right? Your body is constantly running these little investigations and finding tumor cells while they're still individual cells. So if everything I just told you is true, how the heck do you actually ever get cancer? How is that possible? What happens through a variety of like nuanced measures? We don't need to get into the nitty gritty like biochemistry of this, but like those individual tumor cells can, through random chance, develop mutations that basically give them an invisibility cloak, such that now healthy cells that surround it and your immune cells that are floating around in your peripheral circulating blood can't see it. Because normally what happens, that individual tumor cell puts these little chemical flags on its surface that basically say like, I am different, I am foreign, I am not self. And the healthy cells that surround that cancer cell can sense that little chemical flag. They can see it. They can see it being waved in the background. And they're like, that's weird. And so they will send out these little chemical cues into the environment, things like cytokines and many other things. They can send off these little chemical cues to the environment, and they can send this little, it's basically like a little search beacon, a fire alarm, a siren, right? Hence the name of the company, to your immune cells and say like, something's up. I want you to come and run an investigation on this cell. It's acting weird. It's looking weird. I'm suspicious. And your immune cells will swim by. They'll sense those cytokines. They'll sense those signals, those cues, that siren, and they'll come to the site. They'll run an investigation and they'll be like, aha, you're right. I agree. Something's up, and they will chew it up. But if that tumor cell evolves through, again, all kinds of nuanced biochemistry, if it evolves one of those little invisibility cloaks, the healthy cells can no longer sense that something's up. They won't send those chemical cues to the immune system, and the immune system won't come and run an investigation, won't kill it while it's an individual cell. And now it's got the time and the freedom to grow and become that baseball-sized tumor mass, and you're going to get your symptoms. How does our virus work? What we can do is we can actually engineer a virus that's very good at getting inside tumor cells, and it can basically re-deliver those cytokines that normally the healthy cells around that tumor cell would have emitted to kind of like send that signal to the immune system. We can force the tumor to basically kill itself by sending out the signals from within the tumor cell itself. So we can engineer these viruses to go inside the tumor cells, to infiltrate and get inside and release these cytokine signals, these little chemical cues to the immune system from within themselves, which will kill them directly. They're actually very anti-tumor directly, so they can kill the tumor cells themselves. And then B, and that's like the first wave of tumor killing that you get within hours and single-digit days of administering this virus. But then within like single-digit days to weeks, what can happen is as those tumor cells are dying, they can actually secrete and emit those signals out into the local environment to other tumor cells that surround them that maybe didn't receive a virus. And now the immune system can sense those signals. It can be like, I'm receiving a cytokine signal. Like I've been beaconed, I've been called to action. And so you can actually retrain your immune system that, again, is exceptionally good at killing cancer. We can retrain your immune system to basically like be able to re-find these tumor cells that they were previously able to see, but that have been kind of become hidden from these invisibility cloaks. That's a hard word to say. So you can kind of retrain your immune system to be able to re-see these tumors that were previously invisible to them. And then your immune system can come and kind of do the next wave of tumor killing. And so they can come in and through all kinds of different immune mechanisms can come in and kind of like clean up the margins of the tumor from maybe individual tumor cells that didn't receive a virus, but that are still remaining in the tumor. And so you can kind of get these waves of tumor killing after you administer one of these viruses. There's like hours after, where you have this basically the tumor cells will kind of like implode from the cytokines that you've delivered inside of them. And then in kind of the days to weeks after, you have this immune response where your immune system can come in and kind of like pick out the trash and get who's ever left. And again, they're responding to those cytokines that were emitted from the tumor cells that we delivered with a virus. And so you can actually use a virus to do something good, to do something useful. And that's to basically like deliver these already potent anti-cancer drugs that you already make, right? Like you right now, Chris, you have the ability to make the world's best cancer drug. It's called a cytokine. It's inside of you right now. It also does many other things, right? Cytokines are how you fight infections. They respond to allergies and all kinds of things. So it's amongst the most potent things your body makes, right? Everyone remembers right? Like maybe when you got your COVID vaccine or your booster. Remember that fever that you got right after you got that booster where for like 24 hours you were just like incapacitated. You were so hot. Your brain was a mess. Everything hurt. You know, you were 105 degrees, but it was only about 24 hours, right? That
Hmm.
cytokines. So they are amongst the most potent things your body is capable of making, but they have a
Mm.
very, very, very short half-life, and that's on purpose because they're so strong. You don't want them to be running rampant. You can't maintain a 105-degree fever for a month, right? That would kill you, but you can absolutely maintain it for 24 hours,
Mm.
right? So those same proteins your body already knows how to make, already knows how to respond to. It has a checklist, like if I see this, do this. We can basically re-deliver those, but in a very, very, very targeted way inside tumors so that you're not getting that full body fever where the cytokines are swimming around in your peripheral circulating blood and making you feel like you've got menopause or something. Where you're like, why am I 105 degrees? You can deliver those inside of tumors only and have this
Mm.
very, very, very potent targeted response that's anti-tumor but spares the rest of your body.
From a data perspective, what data have you seen in your animal studies that suggests like this isn't just a concept, it could work?
Exactly. So you name it, we've got it. So we've done everything from what we call like in vitro studies. So these are like studies in cells in a dish, right? So this is where we like take actual human tumors from human patients and we test them in a dish and look to see that it works. Check. Then we can do what we call in vivo studies. These are actually studies in animals, and we've done studies in mice and we've done studies in pigs. And in mice, there's like three flavors of mouse studies that we've done. I'll skip all the jargon and the nuance, but we can take like three different classes of mouse studies. We've got mice with mouse brain cancers, and these mice have a fully functional immune system.
Mm.
And we can take like a mouse surrogate of our drug, so like a mouse version of our virus that's delivering a mouse version of our cytokines, and treat those mice and show that we can eliminate their tumors. Then we can take fancy schmancy, and then we're going to get super sciencey here, but without jargon. We can take fancy schmancy humanized mice, where we take mice that have a slightly weakened immune system so that they can accept human tissue grafts. And we can
Mm.
actually take tumors from human patients and we can transplant them into the brains of these mice and grow like a mini human brain tumor, an actual functioning, living, like has a blood supply brain tumor in the living brains of these mice, and
Mm.
then give them the human version of our drug and show that we can eliminate those brain tumors. And we can do that in two different flavors. We can do that with like these like kind of contrived and artificial and engineered human brain tumors that we've made in the lab. We can also do it with like truly what we call primary tumors, where we have taken a tumor directly from a human patient and just cut it up into small pieces and put it into a mouse brain and have done no funky science engineering to it at all. We call those primary tumors. So we can do both engineered human tumors and primary human tumors. And in both cases, we can transplant those into the brains of living mice, grow these human brain tumors, and now treat them with the human version of our virus and show that we can eliminate these tumors. And again, I think we're up to just shy of about 600 mice that we've treated to date, with particularly those humanized mice, where we've shown like across all of these studies over years and years and years, like super reproducible, like 86% of these animals have a complete response, which is like scientific jargon for like they are cured. Like their tumor is gone. It's not just dormant or it's not growing
Mm.
anymore. Or it's not moving. Like it is eliminated and it has not recurred. It has not come back. Like these mice are cured of their brain cancer. And then on the large animal side, we've treated two different flavors basically of pigs. We've treated normal healthy pigs that have no brain tumor at all, just to look at like, do we see any safety signals, any immunogenicity, anything that would make us concerned to go into the clinic, from just a safety perspective? Not like, does the drug
Hmm.
work at reducing brain tumors, but just like, is it safe to administer into the brain, right? Because the route of administration that we're doing is intratumoral. So we're actually injecting these viruses into the tumors in patients. So wanting to inject this into just a healthy brain to see like what are potential side effects that we should be aware of, like do they happen, and if so, what are they, and what's the timeline with which they happen, and like what are the remediation steps that you could take, and these types of things, so that like you kind of know in advance to be prepared for your human clinical trial. And then we've also treated pigs that actually have brain tumors. And so we can actually make these, and the nice thing about pigs, right? These are now large mammals. So like mice are great. They're small mammals. They're cheap. They're easy. They're fast. You can do them in large numbers. But there's something to be said for doing a large mammal. And so the FDA actually requires before you start a human clinical trial, they want you to do, they have a 2 species rule. You have to test your drug in 2 different species. So we've done mice and pigs, and they want one of those species to be a large mammal. So it can be a monkey, a pig, a dog, a sheep, but it's got to be a large mammal. They want something that kind of meets the size of like a human organ, right? And pigs are great because of very similar brain shape and orientation
Mm.
and anatomy to the human brain. So they make a really nice model for brain cancer drugs. And it allows us to actually test like the real rig that we're going to use on human patients, right? Because we need to like make a small, like a little burr hole in your skull. And we go in, right? And we're going to inject the virus into the brain tumor. And so we can actually test that exact setup with the exact tools that we would do on human patients and like test that in an animal that's of the like, kind of like the right size and shape and orientation. And so we've got data from tumor bearing pigs where again we can show like everything is safe, everything is tolerable, everything works. And so now we've got like this whole body of data from like in vitro cells in a dish to these. We also have these fun like 3-dimensional organoids in a dish, if folks know what that means, all the way up to mouse studies. Again, syngeneic allografts, CDX and PDX xenografts, to throw some jargon out there. All of these different kind of humanized mice settings, and now both healthy and tumor-bearing pigs as well. And so we've got like this rich body of data that shows that like it's safe, it's tolerable, it reduces tumor size, and in some cases can actually even like cure and completely eliminate these brain tumors. And so we're as confident as one can be going into the clinic, and super excited to actually start testing this in patients in a phase 1, which is the right. Remember, like the difference between a phase 1, a phase 2, and a phase 3 trial, right? A phase 1, right? You're really just looking for safety signals. You want to make sure like this is safe, it's tolerable in patients, and you want to try to identify like the safest possible dose that you can advance into a phase 2 trial. And then phase 2 is all about like nailing down the dose. Like what is a dose that works? Where like you can see whatever the benefit is that you're trying to find. In our case, right, we're going after tumors, so we want to see tumor shrinkage. Like what is the dose that best shrinks tumors? And then a phase 3 trial is this thing as good as the standard of care? Hopefully it's better. So let's do a head-to-head comparison with whatever the current standard of care is, and look to see whether or not you are actually better. And if you are better, then we want to move you up into the front line and have you become the new standard of care drug. And then after that, right, you file for approval from the FDA, and can start selling and marketing your drug, and patients can go get a prescription for it.
What have you seen in the larger mammal study in terms of ability to kind of reduce or completely kill off the tumor? Um, and in the cases where it's not, what are you learning from that about why it maybe doesn't work?
So what's interesting about the large mammal studies, so very often you don't actually run efficacy studies in large mammals. The
Mm,
large mammals are typically what we call toxicology studies. So that is just to see like are there safety signals that you need to be aware of, and what's happening with your drug? Like where is it going? So even though we've tested it in tumor bearing pigs, the point of that study wasn't to look at efficacy. We took
got
those
it.
pigs down after single digit days. The point of that study was to do tolerability and biodistribution. So you want to see like when we administer like this dose in this volume at this flow rate at this trajectory into the brain, where does it go? Does it stay right where you put it? Does it like leak out a little bit like a millimeter or 10 millimeters? Does it go outside of the brain? Does it go to your toes? Does it go to your eye? Does it go to your fingernails? Does it go to your heart? Like where does it go? So those studies were to look at kind of biodistribution, like where does the virus go,
Mm,
and what were the side effects? Like did the pigs did they get a fever? Were they throwing up? Were they unable to eat? Were they the challenge with all animals? You can't ask them like, how do you feel? Like,
right.
do you have brain fog? Right. So there's some things you can't ask an animal, like how are you feeling, but you can, right? Some things like if they throw up, right, you can see that. If they're shivering, you can see that. If they're really itchy, you can see that. So there's some things you can kind of see and model, but there's some things that you only know until you get into humans. So most of the large mammal studies are actually never to look at, I shouldn't say never, but in our case, in many cancers, it's not actually to look for the efficacy signals. That
Mm.
is still primarily done in a true disease model animal, which is going to be the mice. But in the large animals, you're really looking for safety signals and like the kinetics of the safety signals, the tolerability, and the biodistribution. It's not that you can't see a safety signal, right? Right, like hello, chemotherapy, right? Like, anyone who's ever had cancer or knows someone who has knows that you can be an FDA approved drug that has a toxic side effect. You just have to know what it is going into the trial. And so
Hmm.
that's kind of like the point of these large mammal studies is they want to see like what are all the potential side effects and toxicities and just like immunogenicity signals, these types of things that we might expect to see in a phase 1 trial, so that we can, when you enter a clinical trial, right, you go through an informed consent process. They're like, here's all the things we saw in animals,
Mm.
and the mice, as well as whatever your large mammal was, whether it was a primate, a dog, a pig, a sheep, whatever. Like, do you still want to participate in this trial, knowing that like here's all the statistics, like here's everything we saw, here's all the numbers, here's the side effects that we saw, like here's what we expect is going to happen. Do you consent to participating in these types of things? And so in our case, everything so far has looked absolutely fantastic in the pigs. If I had brain cancer, I would sign up for our clinical trial next year. Like everything we've seen in pigs has been super heartening, that like it seems tolerable. It seems like we don't see any safety signals that would make it that you wouldn't want to advance this into humans. And all of the efficacy studies, like the tumor reduction, the complete responses, the lack of recurrence, all of that data comes from all of those super special humanized mice, where we can test
Mm.
a human version of the virus in mini human brain tumors in the mice.
Very cool. That's amazing. Just to help scope it out, and then I would love to invite Teddy, our kind of senior associate who is leading the effort to decide to invest in this, to talk a little bit more about why we decided to invest. And then also to everyone on the call, please drop your questions in the Q&A section, and we'll work to get to them in the next few minutes. We'll start going through some of those. Just to help kind of scope the opportunity, can you help us understand how big an opportunity this is and where it can lead?
So the really fun thing for us, and one of the reasons why I really wanted to start the company, is that the potential is so high with this universal gene therapy, right? So unlike other gene therapies, where for every single new disease you might want to go after, you're going to need to make a new drug from scratch. We can, like, our lead asset, by the way, is called Siren 101. So the first virus that we're advancing for this first brain cancer. But we don't have to change anything about Siren 101 for the next cancer we go after. We can use the exact same drug, not only the same drug, we can use down to the same lot, right?
Mm.
So we can utilize Siren 101 in the very next indication without having to change anything. So it's basically like, you know, when you think about someone like a classic, like a pipeline on someone's pitch deck or on their website, like here are all the indications that we're going after. All right, and here's how much money it takes to go after each new indication. For us, we can basically short circuit that whole process, because we
Mm.
don't need to start back at day one at discovery. We can reuse Siren 101 now in additional cancers without having to redo just about almost all of those preclinical studies. The only thing we'll likely need to repeat is going to be the toxicology study in a new cancer. But we're not going to need to repeat like all of the like dose escalations and safety studies and these types of things, in order to show that this thing like would work for an additional cancer indication. And so for us, the opportunity, like in the short term, for like the high grade gliomas we're going after first, which is like the most common brain cancer in humans. Number one most common in adults and number two most common in children, universally fatal, no effective drugs, like huge unmet need. There's, I think, something like 22,000 patients diagnosed in just the US market every year with this.
Mm.
Most of those patients, unfortunately, typically die within a year. So it's 22,000 new patients every single year that are getting this unfortunate tumor and really have no effective options. And so the market is huge, even though there is an existing standard of care. It is a standard of care that does not work. So it's the incumbent should should hopefully be feasible to go up against, because it is still a universally kind of fatal diagnosis. And so the nice thing for us as a company is that these trials are very kind of classic and simple in the oncology setting and in the oncology world. If you're talking about, right, the comparison to like a breast cancer, thank goodness we have gotten so good at treating breast cancer that those trials are very long, right? A breast cancer trial might be 8 years for each one of those trials, right? A phase 1,
Mm.
a phase 2, a phase 3, right? They can be very, very long because we have gotten so good at treating it. Versus in the brain cancer space, we are unfortunately still so bad at treating brain cancer, not because our clinicians are doing a bad job, they're doing the absolute best they can, but we just don't have effective drugs that work in this space. And so these trials are very short, because we just don't have
The
effective.
unmet need is so
So
high.
you can actually read out these trials very, very quickly. The other nice thing right? There's 125 at least known primary brain cancer. So different flavors of brain cancer that you can get. There's about 125 of them. Right? High-grade gliomas are the most common, but there's 124 others. Um, and the nice thing is that for the most part, with a little bit of nuance, all of those for the most part, are treated exactly the same. So it's very different than many other solid tumors you might have in like the heart, the lung, the kidneys, these types of things. In the brain, for the most part, everyone kind of gets, again, with a little bit of nuance on the edges, everyone kind of gets the exact same treatment regimen. It's a maximal safe tumor resection if your tumor is in a region of the brain that is resectable. Not all regions of the brain we can go in and cut. You get usually like a 6-ish week course of radiation if your tumor is in a region of the brain that can accommodate radiation. Not all regions can. And then you get a 6-week course of really one of two drugs. Both of them we administer systemically, and they're super toxic, and they're going to make you super sick, and you're going to hate it, and you're going to develop a resistance to them, and they're going to stop working, and then that's when we run out of options. Like that's it. That is the standard of care. For example, in high-grade gliomas, the current standard of care is a drug called Temodar that buys you 2.5 months. Like it's just not, it's not acceptable. Right. And so the nice thing for us is that high-grade gliomas is like the first indication, but we can piggyback and kind of go after like the second most common brain cancer, and the third and the fourth and the fifth in pretty rapid succession. And again, like these are areas where there are no effective drugs. And so you're not kind of up against this big pharma beast that feels impossible. How are you ever going to compete against, Novartis and Pfizer and Merck and GSK and all these groups? And it's like, they don't have anything in brain cancer. And so there really isn't a really challenging big pharma incumbent that you're battling against where it's going to be impossible to kind of like get even 1% of the market. Like 100% of the market is available because there are no effective drugs. And for some reason, the brain tends to be a solid tumor space where there's not a lot of competition, the way that you see with some of the more, I hate to say popular, that's like a gross word, but like the more popular cancers, things like breast cancers, for example, and blood-based cancers. So the opportunity is large. We're talking about single-digit billions for the most part, each one of these cancers in the brain. High-grade gliomas, I think, is just a smidge under 4 billion in just the U.S. market, kind of the annual market opportunity for that. And for the most part, that's going to be the same for each one of those brain cancers. Going to obviously the 125th brain cancer is not going to be a $4 billion opportunity. That is a smaller patient population, but you're looking at like single-digit billions to high hundreds of millions for kind of each of those brain cancers. And that's just the brain, right? We're not limited to the brain. We can go outside of the brain. The advantage for us, though, of staying in the brain in the short term is that, like, man, once you've run one of these trials, like now you can kind of make it cookie cutter. And like the second brain cancer trial it's the exact same trial design with the exact same endpoints, with the exact same drug, the exact same dose, the exact same route of administration, with the same endpoints, with the same kitting, with the same at the same sites. And so it's like you can really make that second trial with that second program just so fast and easy to implement, and needs so much less capital than you needed for even your first program, that it just allows you to really kind of iterate very quickly and just start going down the list and like, let's just try to hit all of them. And in the perfect world, if I had my dream, if the FDA is listening, if I had my dream, our second program won't actually be for an individual brain cancer. You need to do an individual brain cancer for what's called a first in human trial. You need to test a single drug in a single indication. But once you've done what's called a first in human trial, which is the trial we'll start next year, then you don't need to do that for your second program. So in a perfect world, what I'd love to do is what's called a basket trial, where we take basically all comers. Like, do you have brain cancer? Great. like that, you can do a basket trial where you take not individual cancer types, but just like a class of cancers or a class of tumors. And so it can be like, do you have a stage 3 or later brain cancer of any type, meningioma, medulloblastoma, ependymoma, astrocytoma, we don't care what type. And then allows you to just really try to recruit patients much more quickly and get a much broader label. And that would really allow you, with like the minimal amount of capital and the minimal amount of time and minimal amount of effort, to really expand the label really, really quickly. But again, you need that first trial where you've tested just your one drug by itself, no combinations, in a single indication. And you need to kind of like demonstrate to the FDA this thing has legs, it's safe, it's tolerable. And then for your second program, they can allow you to run a basket. And so that's the dream, would be actually to have the second program be a basket trial.
Understood. So I want to bring Teddy up here to just provide a little more color commentary on our analysis and why we decided to invest. And then I will definitely turn it over to some of the great questions that are coming in. And please do continue to drop your questions into here because there's some really good ones. And we always love making sure we get as many answered as possible to really help you think about your investment and make sure you're getting your questions answered. Teddy, good to see you.
Hello, everyone. Nicole, great to see you again as well. Um, yeah, so just wanted to jump in a little bit on kind of what the Kingscrowd Investment Committee, kind of some of the things we were looking at, some of the risks we were looking at, and kind of how Siren has mitigated a lot of those. So just want to start by saying, um, Siren, as Chris said, is one of the strongest companies we've seen in the equity crowdfunding space. And for me, I focus a lot on early stage pharma and med tech deals in the equity crowdfunding space. And really one of the strongest companies I've seen. And we've already hit on a few of these, but just want to kind of lay out the three key risks that we evaluate with pharma companies and how Siren has really addressed those and made us really comfortable with moving forward and investing in this round. Without so the first I would say with any startup, you could say funding risk is always something to consider. A lot of companies in the equity crowdfunding space are raising what I call existential capital, capital runway that they really need to survive. And when it comes to Siren, I'm not sure if Nicole mentioned this, but Siren's backed by two major VCs in Founders Fund and Lux Capital. Founders Fund being one of the most famous VCs in the country, early investors in Airbnb, Stripe, SpaceX, like you name it, they're probably in it. And what was interesting to me is I was looking through Founders Fund's portfolio, and they seem to rarely invest in pharma companies. So the fact that they invested in Siren was some real positive signaling for us and kind of gave us even more comfort in the opportunity and the kind of the de-risked nature of this investment. So with Siren, it's really not your typical equity crowdfunding round, particularly when it comes to pharma, in the fact that, Siren is very well funded, and this equity crowdfunding round is really just an extension of the priced VC round that they ran, and really just allowing folks to invest that, really resonate with the vision and are excited about this. So that's funding risk that Siren has really addressed. Tech risk. Nicole mentioned a lot of this already, but Siren's obviously combining two of these treatments, AAV gene therapy and immunotherapy, to treat cancer. Those have both been around for a long time for different use cases, but in this kind of oncology white space, they're really kind of first movers and really poised to kind of own it. So, as she mentioned, very strong preclinical data in mice and pigs, and kind of a really rare company, rare pharma company in the equity crowdfunding space that is going to potentially enter human trials, early to mid next year, which is usually quite rare. So, and just kind of on the tech in general, it's quite a de-risked investment on the tech side. And lastly, team risk is obviously very important for any startup, of course, but I think even more pronounced in pharmaceutical investing due to, these FDA approval phases. They are very expensive, they're very complex, and startups that are entering those need to really be able to manage burn, hit milestones and kind of deal with anything that might not go to plan, kind of having contingency plans, things like that. And Nicole, I think she a little bit understated said it, but she's really kind of a leader in the AAV gene therapy space, heavily cited scientist. She's a key opinion leader for a lot of different venture capital firms, all these different boards that she's on. And so, not only technically gifted, but I think a very strong communicator in explaining this therapy, which can get quite complex quite quickly. I think she's very good at explaining it to a non-technical audience, and I would encourage everyone to watch her pitch at the All In Summit a couple years ago, where she gave a great presentation to a group of non-technical, in terms of pharma technical listeners, and was really able to get everyone excited about Siren and everything that they're working on. So those are the kind of three main points. And I wanted to ask you, Nicole, in terms of the VC round, the Series A that you raised, kind of what were those milestones when you first raised that? How are those milestones going? And kind of what do you need and what's the timing for a Series B?
So for our Series A, we really had kind of three big milestones that we set out to achieve. The first was to actually get a named development candidate. So prior to the Series A, we were working on a number of things that all universal AAV gene therapies. We needed to like decide like who is the one that we're going to put forward to clinical trials. So actually like make the decision. And right in biotech jargon, we call that like you need to name your development candidate and like put the flag in the ground. Like this is the drug we're moving forward. We're not changing it. Number two was to actually complete all of the process optimization that goes into the manufacturing of the virus that's going to get used in actual human patients in the clinical trial. So there's like years of optimization that you do around like the manufacturing of the virus itself and then actually running the final lot, like what we call the GMP, so good manufacturing practices. It's like jargon for like the cleanest of the clean virus you can ever imagine. It's the one that's actually going to go into human patients. So getting all that process optimization done and then actually running that final manufacturing lot. And then actually doing the study that Chris, you and I were talking about, that very special toxicology study in pigs that is like a prescribed study that the FDA wants to see. So every drug you've ever taken in your entire life, whether it was prescription or over the counter, when it was going up before its first clinical trial, you have to do this thing. It's called an IND-enabling large animal toxicology study. That IND just stands for investigational new drug. And it's this very prescriptive large animal study that happens where you like test 1 million different doses with 1 million different conditions in a bunch of different large animals. And you take all of these animals down and you analyze all the tissues and you look for like every possible signal you can imagine. And it's this like very, very large study that gets run, and you submit like 2,000 pages worth of data back to the FDA. And then at the end of that process, once you've submitted that IND application, that investigational new drug application to the FDA, and they greenlight it, then that is the moment you can open your clinical trial. So our milestones for the A were like get a name development candidate, complete the GMP manufacturing for that development candidate once you had decided which one you were going to advance, and then to run that IND-enabling animal study and get the IND open for the clinical trial. And where we're at on all of those, so we named a development candidate, gosh, I think that was back in like early 2024. So when we put out the press release for that we had decided on Siren 101 and all the component parts that were going into it, and that was going to be our first asset, and then started the process optimization for all of that with our fantastic partners at Catalent. So Catalent is the CDMO, contract development manufacturing organization, for folks who don't know that word. It's just basically like the professionals who are making the virus at scale in a super, super fancy, clean suite. Like if you think in the movies, when you see all the scientists who are in like those complete those white like bubble suits from head to toe, and they're in that clean room and they're doing everything in a very prescribed manner, that's a GMP manufacturing suite for a drug, like you've seen in the movies. So we've actually got all that process optimization done. That all finished up earlier this year. And we're actually like as we speak, as we are talking on this call, Chris, Teddy, and I, the actual virus that's going to be put into humans is being produced as we speak over in Baltimore. So it just finished. They like pulled the virus off the bioreactor, I think like a week and a half ago, and now it's starting the purification process. So we're going through all the purification processes to get that ready, and it will get vialed and released here hopefully in, I believe, September. It will be greenlit, and so then we'll put it in the freezer so it's ready to go. So that will all be done here in about like another like 45 days, and then the IND-enabling animal study. That we had a very big meeting with the FDA back in April. It's called a pre-IND meeting. So like before you run the study, you have one more big like in the weeds meeting with the FDA where you go over all of your data and all the plans before you start the study. And you're like, do you agree? Like we're ready to go. This is the study. Like these are all the endpoints. These are the number of animals. Like this is the deal. And they loved it and gave us a green light. And so we started dosing all of those pigs back in May and June. And it's like you do a staggered dosing because there's so many animals. And then we'll take those animals down throughout the summer and into the fall at different time points. So it's like day 15, day 30, day 45, yada, yada, yada. So we'll take them down over the summer and into this fall and gather all of those tissues and run all of those assays. And that'll take us into the fall. And then we should be able, fingers crossed, as long as there's no delays, should be able to submit that big data packet back to the FDA for review this December. So December 2025. And then as long as FDA timelines hold, fingers crossed, they have normally 30 days to review that. And so hopefully in January, we'll get the green light to be able to legally start opening the clinical trial and start dosing patients. And so we should be able to meet all of our milestones actually ahead of schedule and under budget. So not very often you get to say that.
That's pretty terrific, Nicole. So let's hop on over to some of these questions that we have coming in here. So the first set of questions I'm going to ask you is, how does Siren's AAV-based approach compare to in vivo CAR T strategies? And to add to that, what advantages does Siren's therapy offer over CAR T for solid tumors?
This is a great question. So just upfront, I am like a big believer in all modalities. I want as many shots on goal for patients as possible. I want all of us to win. So like everybody is great and I want everything to advance. Differences. Right, so a virus versus a CAR T cell therapy. If folks don't know, this is just you may have heard of like cell therapies. So instead of using a virus as medicine, you're going to use a cell as medicine. And here what we're doing, there's different nuances of how you can do this, but for the most part, CAR T cell therapies, right? These are where we actually go in, we take cells from you, the patient. We're going to take them out of your body. We're going to do different, depending on the type of CAR T cell therapy we're talking about. The nuances don't matter. But what we can do is actually take cells out of your body. We can re-engineer them to basically retarget them and make them laser focused on going after your tumor, like your personal exact, like you, Chris, your tumor, not like James down the street's tumor, like your tumor. And then we reinfuse those cells back to you so that those cells can home in and try to go re-find the tumor cells that are in your body, because now these CAR T cells have been primed basically with like a seek, hunt, and destroy type strategy to go after those tumors. Can be super effective, wildly effective. The challenge in the brain, and the reason these have not worked to date for really any brain cancer or really almost any solid tumor cancer, there are challenges around getting those to maintain their ability to like have that seek and destroy signal and to not kind of what we kind of call flame out and like stop being effective. In the brain specifically, the big challenge, and this is kind of unique to the brain and not necessarily true in some other organs, but specific for the brain tumor space. The challenge in the brain, and this is one of the reasons why the brain is so hard to treat regardless, not just with cell therapies, but with any modality, cell therapies, antibodies, small molecules, ADCs, viruses, you name it. The challenge historically has been these tumors are notorious for being what's called heterogeneous, or like not of one type. So it's not like every single tumor cell is a perfect clone and it grows out in this perfect spherical tumor mass, and every one of the tumor cells is identical. Quite the opposite. So brain tumors are notorious for being super unique and super different. And like as they're growing, they have all these different, remember those little chemical flags that I was talking about that they put on their surface? They'll have different flags that they put out in different numbers and different orientations, and the flags will be different. Those flags are called antigens. And what that means is that there's not like one thing that you can go after. And so if you make a cancer drug that is targeted for like flag A, but there's flags A through Z present on that brain tumor, you might get rid of 1% of the tumor, but 99% of it remains because they don't have that same shared chemical flag. Or what can happen very often is that brain tumors are also notorious for what's called antigen loss, which is where they just drop their flag. And they're like, well, I don't like that flag anymore. I'm just going to drop it. And I'm going to have a new flag. And I was flag A before, and now I'm going to be flag C. And they can do that. And so what happens if you've spent all this time and money developing a drug that targets flag A, and then the cancer drug or the cancer tumor can just be like, I'm just going to drop it. Like now you have a drug that has nothing to bind to. And so very often what happens with brain tumors is that you have the double whammy problem of the tumors are very heterogeneous and they have no like shared features that you can target with a targeted drug. And cell therapies by their nature are targeted because they have to bind to something, right? If I give you a perfect sphere, it's very hard to grab it unless there's like a notch that you can pull on. And so cell therapies need a notch. They need a thing they can grab onto. And so if you don't have like shared antigens across all of these tumor cells, there's nothing that they can target. And so it makes it very hard to get enough of the tumor cells to die that you have like a meaningful response. And then you have the shared problem that not only are they so heterogeneous, but they can just drop their flags. And so it makes it really hard to make anything that's targeted because it might work 1 second and the next second it doesn't. So the nice thing about a broad virus that is administered inside the tumor is that we don't need it to be targeted on the surface because our targeting comes from the delivery. We can deliver these viruses inside these cells and have them die from within by expressing these cytokines. And so you're not dependent on the tumor being very similar, and you're not dependent on it maintaining these little antigen flags on their surface the way that a cell therapy is. And so I think in the brain cancer space, I think cell therapies are going to be a challenge that may not be surpassable with that technique, and you'll need other techniques. But CAR T can still work phenomenal wonders in other cancers, particularly in blood cancers.
How does the safety profile of your therapy differ from CAR T? And can your therapy offer a more scalable or accessible model than CAR T?
So I can only speak to date in animals, right? Because we don't have human data yet. So I can't comment on human data yet. But in the animal space, like safety wise, right, a viral therapy is going to be very different from a cell therapy. A cell therapy, by its nature, right, it is cells from you, like that we're giving back to you, right? So that's going to have a very different profile than a virus. The nice thing about our virus, and one of the reasons why we chose it, right, folks, if folks remember what we talked about at the very beginning, right? I was not back when I was a professor, I was not an oncology professor. I was not a cancer professor. I was a virologist, like a viral gene therapy professor. And amongst virologists, there's like normal virologists, like 99.99999% of all virologists that you've ever heard of or met or seen in a movie or these types of things, right? They study viruses that make people sick. That's where the money is, right? You study things like measles and mumps and polio and chickenpox and COVID because that's where the need is. I was one of the very, very, very rare, weird virology professors because I studied viruses that don't make people sick or animals sick or plants sick. I study viruses that are just very good at like getting around, but that don't actually make you sick because they're these fantastic little engineerable FedEx trucks that we can do things like engineer to deliver genetic medicines. And so one of the reasons we use this virus, not only to treat little kids with genetic disorders, but now also cancer for the first time, one of the reasons we use this virus is because it is so safe, right? It is not immunogenic. It doesn't stimulate your immune system the way that like a cell therapy can. It doesn't carry diseases. It's not a pathogenic virus. For the most part, your body, I don't want to say ignores it. That's an exaggeration, but for all intents and purposes, relative to something like a normal virus that we think about in a pandemic scenario or something, for the most part, it kind of ignores it. And so it allows you to engineer this very engineerable, you can think about it like software code, this very engineerable thing that you can deliver that your body, for the most part, kind of ignores. And the nice thing for us, this extra added safety feature that we have that's super unique to our virus, is that so the class of viruses that we use that I mentioned before, AAV, which again, the nuance doesn't matter, but adeno-associated viruses. This is one of only 2 viruses that has ever been discovered out of the gazillions of viruses that we've ever found on planet Earth, and there's more to be found we haven't even found yet. There's only 2 that have ever been discovered that have the particular genome conformation, so like the DNA inside of it, the particular genome conformation that this virus has. Right? Everyone remembers from Jurassic Park, right? DNA is a double-stranded helix, right? Normally. But this virus has a single-strand linear DNA. So it's not double-stranded, and many viruses often have a circular genome, and this one's linear. So the combination of single-stranded, linear, and being DNA rather than RNA is super unique. And what it means is it's kind of like a wet piece of spaghetti. So it's unbelievably fragile. I mean, you just like look at this thing and it will shear into a million pieces. So we normally only administer it when we're treating genetic disorders. We only ever administer it to tissues that aren't turning over, right? We wouldn't use it to go after a bone marrow disorder where the cells are turning over all the time because the virus would just instantaneously be lost. It can't withstand that mechanical shear stress because again, its genome is literally like a wet piece of spaghetti noodle. But if you administer it to something that's fast growing, like a tumor, what you get is this beautiful kill switch that you didn't even need to engineer, but you get to leverage because it will get inside, it will release its genome, it will express the payload, in our case, right, these cytokine payloads, and it will express those durably all the way up until the minute that cancer cell dies. And the minute that cancer cell dies, that viral genome will just shear into a million pieces. It's like a firework goes off and you instantaneously lose that virus. So you don't have to worry about the virus continuing to exist in your brain or expressing long-term or going somewhere it's not. It also can't make copies of itself. So you don't have to worry about like spread or these types of things. So it has all these fantastic safety features that, like, fun fact, we didn't even need to engineer in. They are just like the fundamental biology of how that virus works. And so if you know enough about viruses, you can pick the right virus from the get-go. So we can leverage the unique genome composition that this virus has to do something in a really, really safe way, but just in a different way than we had kind of previously ever used it before, and kind of use it in a cancer setting now rather than in a genetic disorder setting.
Two last questions here, and we'll just hit on them real
And I'll
quick since we're
answer
coming to the top of the hour. So in terms of patents, this individual is just wondering, no future surprises from unknown biotechs claiming them. And
Yeah,
then
no, we own the white space. We've got it. We've got a huge patent portfolio, and it's all us. Both methods of use and composition of matter.
Terrific. And then the second one is just in terms of where you go next outside of brain cancers. It sounds like there's plenty to tackle there already, but outside of it, prostate cancer, lung cancer, is there a linear path that makes sense for you to go to next? What are your thoughts on that?
The brain is definitely, in the short term, it is our focus because we can move so quickly into like the second kind of follow-in indication. And again, the goal would be to queue up like a basket trial, but we can absolutely go outside the brain into other solid tumors. So the priority would still be solid tumors, so not the blood-based tumors, so still solids. And there we would just kind of be going down the matrix of like, what are other solid tumor indications where there's still a high unmet need, where there's not huge incumbents, where there's not an existing standard of care that works, and where we think Siren 101 can still have like a big effect. And so that's where one of the places where we're using AI tools to basically take all of our in-house data with all of the publicly available data that you can get from like all of the cancer consortia, all of the published papers, all of the data on clinicaltrials.gov, all of the patents that are available. And you can take all of this data, map it on top of all of your in-house data that you have with your drug, and be like, okay, what are other solid tumor cancers that pre-treatment have the same signatures, the same genome composition, the same proteome composition, the same epigenome? What are other tumors that look like, not in terms of like physically look, but like at the genomic level, that have a signature that looks like the cancers in the brain we've already successfully treated? And we can use that to kind of guide what are the animal studies we might want to do now to kind of figure out what are the other solid tumor cancers that are likely to be highly responsive to Siren 101. So that way we can kind of better prioritize the dollars that we have and use our capital more efficiently to go after the next like solid tumor cancer outside of the brain, in kind of the fastest route possible. And those studies are ongoing.
Understood. Well, thank you so much, Nicole. This was a wonderful, wonderful session, super helpful for diving in further on what Siren is all about. For those who are listening, thank you for joining us today. You could check out our report, which I believe we've unlocked, if you check in the chat section, and we'll be sending a follow-up as well. If you have questions, thoughts on how we can make this even more helpful for you, please let us know. We always love to hear from you all. And if you're interested, certainly go check out Siren and invest on Wefunder today. Thank you so much, everyone, for attending. Thank you, Nicole. It's been wonderful.
No worries. This is great.
Have a wonderful day, everyone. Take care.